Welcome to the continuation of our lecture on Autacoids. The first Autacoid that we are going to discuss is histamine. For the outline of this presentation, the first part of this presentation will talk about the introduction to histamine. We will also talk about the biosynthesis, the metabolism, the distribution and storage sites, as well as the release of histamine. We will also going to discuss the pharmacology of the histamine receptors. For the learning objectives, this presentation aims to describe histamine, its biosynthesis, storage, metabolism, and release. And it also aims to identify and describe the different histamine receptors, their location, effects of activation, agonist and antagonist. So how do we define histamine? So histamine is named after the Greek word histos, meaning tissue, because it is present in tissues and also because it is an amine. Histamine is an autacoid that is present in man and animals, venoms, and other zootoxins, bacteria, and certain plants and vegetables. Dietary ingestion of these food products will not contribute to body histamine because whatever is absorbed is metabolized in the liver. Intestinal bacteria can also synthesize histamine, but it is also degraded in the liver after absorption. Histamine is involved in inflammation, anaphylaxis, allergies, and certain types of drug reactions, and it regulates gastric secretion. Chemically, histamine is beta-imidazole ethylamine. For its biosynthesis, histamine is a monoamine synthesized from the amino acid histidine through a reaction catalyzed by the enzyme histidine decarboxylase or HDC, which removes carboxyl group from histidine, yielding histamine. This diagram shows the histamine metabolism in humans. Histamine is metabolized via two pathways, predominantly by methylation of the ring, followed by oxidative deamination, as shown in this figure. Secondarily, histamine is metabolized by oxidative deamination and then conjugation with ribose. After synthesis and release from each storage sites, histamine acts at its target and is rapidly metabolized through two primary pathways. The first is, again, oxidative deamination. Oxidative deamination leads to the formation of the compound imidazole acetic acid, while methylation, which predominates in the brain, leads to the formation of N-methyl-imidazole acetic acid, or the N-methylhistamine. So both metabolites are inactive and subject to further biotransformation. The methylation is the major degradative pathway and the oxidative deamination is the minor pathway. For its distribution in storage sites, histamine is widely distributed in tissues and its concentration and rate of synthesis varies greatly from tissue to tissue. Histamine is synthesized primarily by mast cells, basophils, histaminergic neurons in the basal ganglia of the brain, and the enterochromaffin-like cells, ECL, in the stomach. These cells produce large amounts of histamine and are thought to be the major histamine-producing cells. The primary tissue sites storing histamine are the GIT, the skin, and the lungs. Food in vagal stimulation can release histamine from the stomach mucosal and enterochromaffin-like cells. The released histamine then initiates gastric acid secretion. Allergic responses in the skin and the lungs are due in part to histamine release from mast cells. Mast cells are the primary cells that store histamine, where it exists in the complex with heparin sulfate and chondroitin sulfate in storage granules. Histamine are also found in the CNS, where it may act as a neurotransmitter. Many venoms and insect stings contain histamine as well as other biologically active substances. Histamine is found in the digesta, where it is formed in large part by the bacterial action. This histamine normally does not reach the systemic circulation since it is metabolized by enzymes in the gut wall and the liver. For histamine release, there are various mechanisms whereby the histamine can be released from its storage site. The first mechanism that we are going to discuss is the immune release. So this involves the sensitized mast cells or basophils that are coupled to IgE antibodies. So when these cells will be exposed to the proper antigen, the mast cells will degranulate, thereby releasing histamine and other Autacoids. So this is also referred to as the type 1 IgE-mediated hypersensitivity reaction. The diagram shows the pathophysiology of the type 1 hypersensitivity reaction. So let's start with our APC and our allergen. APC, or the antigen presenting cells, will recognize the allergen and it will present it to the naive T cells by way of our TCR or the T cell receptor. The T cells will now differentiate into the Th2 cells. The Th2 cells will release the interleukins or the IL-4, IL-5, and IL-13. These interleukins will stimulate the B cells to produce IgE. Next, the antigen-specific IgE will now bind to the mast cells and the basophils. Now, we have here the presence of the IgE receptor in the mast cells or in the basophils. Subsequent exposure to the same antigen, so we have here the presence of the allergen. The allergen serves as the antigen. So subsequent exposure to this particular, the same antigen will lead to the degranulation of our mast cells and basophils to release the mediators of inflammation, including the Autacoids, histamine, leukotrienes, prostaglandins, and the platelet activating factor. Another factor that will enable the release of histamine from storage sites is the drug-induced release of histamine. So certain drugs, which are collectively known as histamine liberators, including morphine, polymyxin, atropine, lidocaine, and penicillin, to name a few, and also their vehicles are capable of releasing histamine, but this release does not involve degranulation or mast cell injury. These drugs displace or compete with histamine for the binding sites with heparin. So we have here a long list of drugs that are capable of releasing histamine or inhibiting diamine oxidase. So as you can see here, we have a number of agents or drugs that are capable of releasing histamine, from muscle relaxants down to analgesics, to local anesthetics, antibiotics, and other drugs. So this diagram was adapted from Mainz and Laura, Mainz et al. 2007, and this was published in the American Journal of Clinical Nutrition. Another stimuli that will enable the release of histamine from storage sites are the plant and animal stings. So the plant and animal stings are capable of releasing histamine, which is an important component of the physiologic reaction such as erythema, pain, itch to these stings. We have here a diagram to illustrate the steps involved in the immediate hypersensitivity reaction after an animal sting. So we start with number 1. So a person is said to be born with a predisposition for allergies. When that particular person will be stung for the first time, this event will potentially induce the formation of IgE. So what is IgE? IgE is a type of antibody that will bind to our mast cells. So as you can see here, we have here our mast cells and we have here our IgE. So when, for example, a particular person will be stung for the first time, the IgE will be formed and some of the IgE will bind to our mast cells. When that particular person will be stung for a subsequent time, when that particular sensitized person will be stung for a subsequent time, the allergen from the animal sting will be deposited to the blood and it will reach the IgE. The IgE now will form a cross-link with the injected allergen. So the injected allergen will bind to the adjacent unit that was previously bound to the mast cells. So as you can see here, we have here our injected allergen and we also have here our IgE. The IgE is bound to our mast cells. The injected allergen from the animal sting forms a cross-link with our IgE. So the mast cells with cross-linked IgE is important to release histamine and other chemicals that cause the allergic reaction. So in the presence of a cross-linked IgE, this will be an important stimuli for the release of histamine from our mast cells. So after this, systemic allergic reactions can occur very rapidly and can be life-threatening. And because this is life-threatening, they may require immediate medical attention. Another mechanism whereby histamine can be released from storage sites is through physical injury. So an example of physical injury that can induce the release of histamine are heat, cold, or trauma. And these factors can disrupt the mast cells, thereby releasing the histamine. So we have here a diagram to illustrate the role of physical injury, particularly a wound, for as a stimuli for the release of histamine from its storage sites. So we have here the presence of mast cells, and we also have here the presence of a physical injury, a cut or a wound. The mast cells here will detect the injury to nearby cells and it will release the histamine. Histamine is important to initiate the inflammatory response. In letter B, the histamine will increase the blood flow to the wound site and it will also increase the vascular permeability, allowing fluid, proteins, phagocytes, and other immune cells to enter the infected tissue. So these events result in the swelling. So as you can see here, there is a change in the structure as well as the size of the area. It will result in swelling and reddening of the injured site and increase blood flow to the injured site, causing it to feel warm. We also have here a diagram showing the histamine-producing cells and the stimuli that trigger the histamine release. So this was adapted from Wang, 2018, and this was published in the Frontiers in Immunology. So as you can see here, we have here our major histamine-producing cells, and we also have here our activation signals. These activation signals are important stimuli for the release of histamine from these major histamine-producing cells. So histamine is synthesized primarily by mast cells, basophils, histaminergic neurons of the basal ganglia of the brain, and also the enterochromaffin-like cells, ECL, in the stomach. So these cells produce large amounts of histamine and are thought to be the major histamine-producing cells in the body. They continuously synthesize histamine, which is then linked to the carboxyl group of heparin and stored in intracellular granules until the cells receive the appropriate activating stimulus. So you can see here the presence of the granules in these cells that contains the histamine. Upon external stimulation, these cells degranulate, releasing the stored histamine. So the stimuli that trigger histamine release by these major histamine-producing cells have been extensively reviewed. The antigen cross-linking of antigen-specific IgE bound to the high-affinity IgE receptor on the mast cell and basophil surface is considered to be the most robust stimulus that triggers histamine release by these cells. So we have here, the presence of these factors are important for the activation of these cells, the major histamine-producing cells, for it to be able to release the histamine. Aside from these cells, we also have the minor histamine-producing cells, such as the dendritic cells, T cells, macrophages, neutrophils, and the epithelial cells. Let's now proceed to the pharmacology of the different histamine receptors. So the effects of histamine are due to the activation of histamine receptors, which are classified as H1, H2, H3, and H4 receptors. The H1 receptors are mainly located in the smooth muscles, particularly in the intestine. It is also located in the bronchi, the blood vessels, the uterus, the brain, particularly in the chemoreceptor trigger zone, and the afferent nerve endings. When these receptors will be activated, it will induce the contraction of the intestines, as well as the bronchi and the bronchioles and the uterus. For the blood vessels, there will be a dilatation, increased capillary permeability, and edema. There are considerable species variation in the sensitivity of the bronchial smooth muscle to contraction by histamine via the H1 receptor. The guinea pig bronchi are considered to be the most sensitive to the contractile effect of histamine to the bronchi. But the bronchi of the rabbit, dog, goat, calf, pig, horse, and human also contract. In contrast, histamine relaxes the respiratory smooth muscle in the cats and sheep. So those are some of the species differences for the effect on the activation of the H1 receptors. The agonists of the H1 receptors include histamine and 2-methylhistamine. For its antagonist, so these are the drugs that are antagonists to the H1 receptor. So these include mipyramine, promethazine, diphenhydramine, pheniramine, astemizole, and the cetirizine. Next is the H2 receptor. The H2 receptors are mainly located in the gastric parietal cells, heart in the brain, cat trachea, sheep bronchi, and the rat uterus. When the H2 receptors will be activated from these sites, it will result to the stimulation of the gastric acid secretion and the relaxation of the vascular smooth muscle, resulting to vasodilation, as opposed to the effect of the H1, which is contraction. There will also be an increase in the rate in the cardiac output as well as CNS excitation. For the agonist of the H2 receptor, so this include histamine and dimaprit. And for its antagonist, so this includes cimetidine, ranitidine, nisatidine, and famotidine. This is the H3 receptors. So the H3 receptors are mainly located in the CNS. H3 receptors are located presynaptically on neurons and inhibit neurotransmitter release. For the effects of the activation, so when these H3 receptors will be activated, this will result to the inhibition of the other transmitters, excitation, and autoinhibition in the CNS. For the agonist of the H3 receptors, so this include the histamine and the alpha-methylhistamine. For its antagonist, this include thiopyramide. For the H4 receptors, so they are mainly expressed in the hematopoietic cells, and one of its effects is the mast cell chemotaxis. We have here a table showing the different receptor subtypes of histamine, their G protein coupling, and the signal transduction. For the H1 receptors, so they are considered to be GQ protein, phospholipase C, and it mediates its effects via increasing and activating the protein kinase C. We also have the H2. The H2 induces or mediates its effect via the stimulation of the GS-coupled H2 receptors, activating the adenyl cyclase and increasing tissue CAMP levels. For the H3 receptors are coupled to GIO protein, and it mediates its effect via the inhibition of the adenylate cyclase. For the H4 receptors, it mediates its effect via also the inhibition of the adenylate cyclase. This diagram shows an overview of the main functions of the histamine receptors. So we have here the four histamine receptors, H1, H2, H3, and H4. So this H1 receptor here are said to be important for its role in allergic inflammation. H2 receptors are important for its role in gastric acid secretion. H3 for its neurotransmission, and for H4 for immunomodulation. So the These H1 receptors, for example, have this particular function. So for example, it alters the vascular permeability, it induces extravasation, edema, addition of molecules, and the migration of the different inflammatory cells. For the H2 receptors, it regulates the gastric acid secretion, and it has also some role in the immune cell differentiation. For the H3, its main role are in the regulation of the neuronal histamine turnover. And for the H4, we have the immune cell chemotaxis and the immune response, as well as the inflammation.